Hi all, welcome back to Microbiology Lectures. This lecture we're going to discuss the membrane of bacteria and archaea, and we're going to discuss the cytoplasmic membrane and a little bit on how it regulates what enters and exits the cell. Okay. So to start off, the cytoplasmic membrane is very similar to the plasma membrane or R membrane that we have, or eukaryotic membrane that we have. Again, we often talk about the membrane in the sense that it's two pieces with a hydrophilic head, which is made out of phosphate group. And attached to that hydrophilic head is a neck, which is kind of a glycerol neck, and then fatty acid tails. So two fatty acid tails or chains that come off of it, which are hydrophobic. Okay? And so that phospholipid bilayer, so it's two layers, the hydrophilic heads are going to be pointing out. Okay, like you can see here, and the hydrophobic tails are going to be pointing in. Okay, so hydrophilic head, phosphate, and with a glycerol connection to the fatty acid, and then hydrophobic tails, which are these fatty acid chains that come off. Okay. Because these membranes are arranged in which they are and composed of the material that they are, it requires a lot of proteins to be embedded in the membrane for things to travel through. Okay, so things that are charged or molecules that have a charge or molecules that are too large will often not be able to enter through the membrane. So they have to enter through some type of protein. These are transmembrane proteins, proteins that go all the way through the membrane and allow for access or ports to go through. Some are facultative in the sense that they just allow material to flow back and forth. Some require energy through ATP or maybe electrons to pump things across, and then others might change the material as it comes across or bind it to something that when it comes across. Okay, and we'll look at those and we'll talk about those as we progress. Okay, so again, the proteins will serve many different functions. Okay, some are selective gates, so they might be very specific to a single thing. Like you might have a protein that only allows sodium to enter the cell, or glucose to enter the cell. Okay, they can sense environmental changes and conditions, so you can have heat-sensitive proteins or saline-sensitive proteins that might indicate to the cell that it's time to release more water or conserve water, close off your aquaporons, this kind of thing. And then some can be enzymes, and we'll talk more about this, especially when it comes to prokaryotes. They don't have membrane-bound organelles, so when we're talking about energy generation, like the electron transport chain, those proteins happen inside the membrane. That would mean that things are being pumped out and then back in to the cytoplasm. Okay, and we'll talk about that when we talk about energy. The membrane itself, we often consider a fluid mosaic. Okay, so that means that the membrane is not rigid, but it can flow, it can change shape. And this is very important, especially for prokaryotes, because a lot of water comes rushing into prokaryotes in many situations, and they have to have that flexibility. They have to have that ability to grow and shrink quite often. Okay, and the proteins can drift throughout the lipid bilayer, so they can move around and then sometimes they don't move around, and maybe it's a biochemical pathway in the sense that you might have proteins lined up together, and one protein dictates the substrate, its product dictates the substrate for another protein. We'll talk about this as we progress. Okay. A couple cool things and interesting things about the differences between archaea phospholipids and eukaryotic phospholipids is, again, they have this phosphate head. Both groups have phosphate head, so no matter what the domain is, they have a phosphate head, and they have fatty acid tails. But how that head is linked to that those fatty acid tails is a glycerol, but in archaea it's a different type of glycerol, or at least the linkage between the glycerol and the fatty acid tail is a different type. It's an ether linkage versus an ester linkage. When we look at eukaryotic and bacteria phospholipids, they have ester linkages that link the phosphate or phosphorus head to the fatty acid tail. Okay? And there are many hypotheses around this, but the one that has probably the most evidence for it, and at least for why, is that these ether linkages seem to be a little more stable in extreme thermal environments than the ester linkage. So that might provide a little bit of stability in really hot environments. The other thing is that you can probably see here is the fatty acid tails have branches, and you don't see that in eukaryotic or bacteria phospholipids. So again, probably the evidence points to that branch tail also gives them some advantage in extreme temperatures, and doesn't allow for the tails to kind of either kink and form extra bonds, but it also doesn't allow them to separate, and it really keeps them a little bit more rigid, so you don't have a flow or a membrane that's too loose, especially when the temperatures heat quite a bit. Those membranes can flex too much and create like open regions that don't provide any support. Okay? At least that's where the evidence suggests right now on why there is this difference between these two groups when it comes to that phospholipid bilayer. There's another group, or there's another big difference in phospholipid bilayer in archaea versus eukaryotic and bacteria, and we'll come back and talk about it a little bit more, but there are archaea phospholipid bilayers that they're connected. The fatty acid tails of one phospholipid is connected to the other, and so there's no space in between them, and we also think that gives them an advantage in extreme temperature environments, but we'll talk about that later. Okay, so the cytoplasmic membrane is a selective permeable membrane. Okay, so again, like I said before, it's going to allow certain small molecules, non-hydrophobic molecules, or small hydrophobic molecules, but hydrophilic molecules are often not allowed through. And then water a lot of times will pass freely, either through aquaporons or just through the membrane. Okay, so lots of eukaryotic cells have aquaporons, and then there are quite a few bacteria and archaea cells that will have these specialized proteins, which we call aquaporon, that allows water to just flow through the membrane. So again, gases, they tend to pass fairly easily through the membrane. Small hydrophobic molecules will pass through, okay, because a lot of the interaction happens here with these hydrophobic fatty acid tails, okay, and so hydrophilic molecules, they typically can't get through the hydrophobic region, so they don't make it through. And then, of course, large molecules, macromolecules, Even ATP, amino acids, these other things, they don't make it, and charged molecules don't make it through either. Okay, so it's very similar to the eukaryotic membrane, like what gets in, what doesn't. Okay, and you can see by the structure there. I mean, at least for bacteria and eukaryotes, it's basically the exact same. So the permeability or the selectivity of the permeable membranes nearly the same between those organisms. All right, so that kind of brings us to simple diffusion. This should be really review for most people. Okay, simple diffusion is just moving from high concentration to low until equilibrium is reached. But when we say until equilibrium is reached, just realize that, so this is high concentration is going to move to the low concentration. So let's say this is outside the cell and it's moving into the cytoplasm. Okay, that material goes through, moves to the cytoplasm, but it's showing you that the material is not moving back and forth, but it still moves back and forth quite often because nothing is completely stable. So the extracellular fluid in this case would be changing, the cytoplasm might be changing, and so material is constantly going back and forth across the membrane if it has the capabilities of doing so. One key component with simple diffusion, though, is the concentration on the one side, okay, will determine the speed at which the reaction or the diffusion process happens. So the higher the concentration or the greater the difference is, the faster the rate of diffusion. So often we will indicate a time to diffusion, okay, so the time it takes for that material to diffuse across the membrane. And that plays a role in lots of different things, antibiotics, drug transfer, that kind of stuff. Okay, osmosis is similar to diffusion, except for it's basically water, and it moves often in what we would intuitively think the opposite direction as diffusion. So in other words, water moves to regions that have a higher concentration of solutes. Okay, so if you have, let's say you have salt molecules over here and a lot of salt molecules over here, water is going to move in to try to make the two concentrations of solute molecules as equal as possible. Okay, so a hypotonic solution means less solutes. A hypertonic means more solutes. Okay. So again, water diffuses from high water concentration or low solute concentration to a low water concentration or high solute concentration. Okay, and often we think of that as kind of being the opposite of, because we're normally talking about the solutes that are in concentration. Okay. It flows from a hypotonic or low solute concentration to a hypertonic high solute concentration. Okay, and then we normally will say that if there's no net water flow or no real difference, so again, just like diffusion, water will be moving back and forth. Even when it's isotonic, water will be moving back and forth, and that's what this picture is indicating. This red blood cell is an isotonic solution, indicating that water flows in and water flows out, and it maintains a constant, or as constant as possible, solution on the inside and the outside. So the solutes on the outside and the inside are nearly equal, and the water moves back and forth to keep that isotonic solution as stable as possible. Okay, so why should we care? Why do we care about osmosis and diffusion, and what does it mean to prokaryotes? So the one piece that prokaryotes got going for them is that they're single cellular, so just a unicell package that really all of life, everything they need can be done with inside that one package. It's not required to have other cells sending you stuff or anything like that, like in a multicellular organism. I mean, it's all in there. But because it's all in there, a lot of single-celled organisms will store material inside their cell, in their cytoplasm. They'll store nitrogen and phosphorus and phosphates, and they'll build it up inside that cell, which often when we see them in an environment, they are in an environment where they are hypotonic relative to the environment. So water is rushing in to the prokaryote at all times, basically. Okay? And that's really where that flexible membrane comes in, and the fact that your cytoplasmic membrane has that ability to flex. And so the water rushes in, it can flex, and it doesn't burst. But if a huge amount of water is coming in, that's where the cell wall comes into play. Is the cell wall is rigid enough that it won't allow the cytoplasmic membrane to rupture. Okay? Take like a human cell, and we were to take a human cell and drop it into a hypertonic solution. The water would rush in, and because we have no cell wall, eventually the cell membrane would split and rupture and destroy the cell. In the case of bacteria and archaea, that doesn't happen very often because the cell wall is rigid and it prevents it from rupturing. Right? But you got a similar situation, or I guess a little bit different, but still a benefit of the cell wall when you go into a hypertonic solution and all the water is leaving to try to balance the solutes on the outside with the solutes on the inside. So the water is leaving, okay, and the membrane will pull away from the cell wall, but because the cell wall is rigid and doesn't flex like the cell membrane, it will still provide protection for that cell membrane, for that cytoplasmic membrane. It doesn't allow things to poke holes in that cytoplasmic membrane when it lacks water, or when it shrink off of the cell wall. Okay? So water flows into cells when the cytoplasmic concentration solution is hypertonic. Okay, so if the solution inside is hypertonic, water's flowing in. If the solution outside is hypertonic, then water's flowing out. Okay, again, the cell wall prevents the cell from bursting, and in regions where the cell membrane is pulled away from the cell wall, it protects the cell from damage, the membrane from being damaged. Okay, next time we're going to talk about energy transfer and how the cell membrane determines how energy is transferred.